Hello and welcome to this podcast. And in this podcast we're going to describe the last two types of horizontal gene transfer. In the last podcast we talked about conjugation as the first type of horizontal gene transfer. In this podcast we're going to talk about transduction, and then we'll close with talking about transformation. But let's start with transduction. We've talked a little bit about this type of horizontal gene transfer when we talked about the Hershey-Chase experiments. We just didn't at that time call it transduction. During the transduction, we begin by taking a donor cell. So this is our donor. And this is the genome in it. And let's say it has a gene on it, like methionine plus. So it's prototrophic for methionine. It can make its own methionine. And then let's take a recipient cell over here, also E. coli, and also with a genome. And let's say the donor cell here, the recipient cell is met minus. And let's just draw the gene on both of these as this little black line. And for fun, right, let's just make sure we say these are both F minus. So we know they're not going to be able to conjugate with each other. So how are we going to get this methionine gene from the donor cell to the recipient cell? One way we can do this is by taking a bacteriophage. And often we will just call this phage for short. And a bacteriophage would look something like this. It has a hexagon type shape to it and a structure like so. And it'll attach to the outside of the E. coli cell. As we mentioned with the Hershey-Chase experiments, the bacteriophage has its DNA in here. It injects it into the E. coli cell. Once inside of this cell, it can begin to digest the genome into fragments. And of course, it will still also have the phage genome in here as well. The phage genome contains the genes necessary to make new phage particles. And I'm just going to draw them quickly like so. We'll make a lot of them, in fact. Some of them will contain the phage genome. It will incorporate some of these fragments of gene. However, some of them will pick up the donor or the E. coli genome. And I didn't write it here, but one of these fragments is going to have our methionine plus gene on it. And so you could imagine how there could be a bacteriophage that could collect that gene. At some point what will happen is this E. coli here, the donor strain, will lyse. It will begin to rupture. In that process, all of these phage particles will exit. And they will be free to now infect new E. coli cells. Now the ones that contain the phage genome, they will go on to infect a E. coli cell and complete this same process over and over again. However, those phage that contain the genome from the bacteria will go on to infect other cells. And if it happens to contain our methionine plus gene here, if this cell now, or this bacteriophage, we're now to come up here and infect this cell. In fact, let's go ahead and have it do that. And I'm going to draw it large. But it contains that bacterial genome plus that met plus gene. It will then insert this genome, which originally came from this E. coli over here, into this cell. Now what happens is a recombination event. I'm going to draw this E. coli. I'm going to draw it a little bit bigger too. We're blowing this out a little bit so we can see it. And this original one that was present all along, it's right here, and I'm going to draw it like so. And I'm going to write minus under it, just so we remember that it's a met minus cell. This genome from the bacteriophage is now right here. And it contains the MET plus gene. What happens now is something called recombination, in which the MET plus gene from the DNA that was inserted by this bacteriophage now recombines, or you could say is introduced into the bacterial genome. In doing so, it displaces the minus gene. So you end up with an E. coli cell from this recipient cell here that now has a genome just like it had before, except now it has MET plus in it. It has now become a prototroph for MET plus. The gene that was here, the minus gene, when it's displaced, it's just degraded. So it's not like hanging around in the cytoplasm anywhere. It's just gone. I'm going to go over this again quickly to point out the highlights again. Remember, we started off with two cells. One that was prototrophic for methionine, that is, it was MET plus, and one that was auxotrophic for methionine, that is, it was MET minus. We introduced the donor strain with the MET plus gene in it, with a bacteriophage. When this bacteriophage infected this donor E. coli cell, it degraded the genome of the E. coli, as we see here with these fragments. The genome from the bacteriophage that's here can now synthesize proteins to make new bacteriophage particles. These particles can now take up various pieces of DNA within the cytoplasm. Sometimes they'll take up bacteriophage DNA, and when they do that, it makes a bacteriophage, such as this, that can now go on and reinfect other E. coli and keep this cycle going forward. These bacteriophage that took up these genomes, these pieces of genome from the E. coli, can now go on and they can still infect the cell. As we see here, bacteriophage attaches to the outside of this E. coli. It inserts this gene, because this is what the bacteriophage took up, it inserts the methionine plus gene that it got from over here. Once in this recipient cell, the methionine plus gene, because it shares a sequence with the minus gene here, will recombine with it, displacing the minus gene, and it will be degraded then. And then this positive gene, the MET plus gene, now integrates inside of the genome. So this E. coli, which was initially MET minus, is now MET plus, meaning it can grow in minimal media. So this is one explanation of how genes can be transferred from one cell to another cell without those two cells coming into physical contact with each other. Let's talk about the third method of horizontal gene transfer in bacteria. And this third method is called transformation. And this is similar to what was described in the Griffiths experiment and in the Avery, McLeod, and McCarty experiment, where they show transformation of rough bacteria into smooth bacteria. So let's start off with a cell. So let's start off with an E. coli cell. We'll draw it like so. And let's draw the genome inside of it. And let's say this time we'll pick a different amino acid gene. And let's say it's His minus. This is going to be a recipient cell. So it's His minus. And if this cell is going to become His plus without coming in contact with another bacteria, say through conjugation, and there are no bacteriophage around, how's it going to do it? With any given community of bacteria, you may find some cells that are His plus. Again, let's just say these are both F minus. So they can't undergo conjugation. So how are we going to transfer this His plus gene over here without conjugation and without a virus? Well, over time within any given population of these E. coli, some of these cells are going to die for various reasons. It's the sad truth, but it happens. And so the membranes are going to burst open or just split over time. And when that happens, the chromosomes also begin to fragment. And when that happens, you're going to get random pieces of this genome released. Some of these genomes are going to contain that, some of these fragments I should say, are going to contain that His plus gene. Now how is this His plus gene that's on this fragment going to get inside of this cell. Turns out there's a receptor here. All of our cells have all kinds of receptors on them, and all these receptors this is what it kind of sounds like. They receive specific molecules. In this case, this receptor will accept certain pieces of DNA. Now this fragment's now going to come over here, and I'm going to draw it as it really would appear as a double-stranded piece of DNA. And as it binds to this receptor, something interesting kind of happens here. So I'm going to draw it down here as the kind of the next stage. As this double-stranded piece of DNA is lined up here and attached to this receptor, one of these strands is degraded by a nuclease. And then all that's left is the single strand. This single strand now can feed nicely through this receptor. And again, you have your genome here, the recipient genome, which is His minus with this line here. And now this single strand here, which has the His plus gene on it, will now undergo homologous recombination with the recipient genome, that's His minus, just like we talked about with the bacteriophage. In doing so, it receives this part of the gene, the His plus gene, and places it where the His minus gene used to be. And so the end result here is a cell like such with the bacterial genome in it, but now it contains the His plus gene from this degraded one over here. The process of it going from a single-stranded piece of DNA and combining and then becoming essentially a double helix again is not important for the purpose of this class. You should just know that it is a single strand that enters in here, and that single strand recombines with this bacterial chromosome, and when it recombines, it now forms a stable gene, new gene, in this case histidine plus gene, in this cell. And now we've gone from a His auxotroph to a His prototroph. So that's all I have for this particular podcast. Let's go ahead and close with a quick summary. In this podcast, we talked about the last two methods of horizontal gene transfer. We talked about transduction. So you should be familiar with transduction. You should be able to draw that out. And remember, transduction dealt with the bacteriophage. And then next we talked about transformation. And transformation involved bringing in DNA that is found outside of the cell and bringing it in and recombining with the genome. In both these cases, this is how we introduce genes from one bacteria to another bacteria without actual physical contact between those two bacterial cells. Alright, well that ends this podcast. If you have any questions, please let me know and I'll be happy to help in any way I can. I'll see you in class. Bye.